Hey everyone. Tonight we're going to be going through the anatomy of a flowering plant lab. And so what I'm going to do is I'm going to share my screen. Um, and then you'll kind of be looking at me as we go through the first part of this, just kind of prepping everything. And then what I'm going to do is I'm just going to angle it down. So since this is a dissection lab, um, everyone can kind of see everything. But I just want to kind of walk through the beginning of this first. As always, feel free to read through the overview, but really the outcomes of this, we want to recognize the reproductive structures of a plant. We want to understand the anatomy of a flower and the function of the flower parts. We want to match flower types with pollination methods, and we want to determine flower types. So the time requirements, we have about 10 minutes of prep, which is just going to be gathering your materials. This lab should have come in the box in your lab kit. If you don't have it, just let me know. And then activity one, we only have one activity here, but it's the dissection of the four flowering plants that are provided. And that they say takes about 60 minutes. I think that's a little overzealous in terms of time, but we'll see. Yeah, so just a key for what we're going to be dealing with. So background. So as normal, take a couple minutes, pause the video here, read through the background. The background's, I think, like 3 or 4 pages, but it pretty much walks you through asexual reproduction and sexual reproduction in plants. Importantly, the part to really pay attention to are the anatomy or the actual parts of the flowering plant, or what we think of as the flower. So everything included within the pistil and everything included within the stamen, the petals and the sepal and the peduncle. Those are going to be important. And just kind of this idea of, the stamen, particularly the anthers releasing sperm or pollen, and the stigma, which goes down through the style into the ovary, as being the receptive aspect of the flower to the pollen. All right, and then it kind of gets into types of pollinators, and how pollination methods can directly affect which flowers a particular flower can mate with. So take some time, go through this. And when you get down to the materials, unpause and we can pick it back up. So go ahead and pause the video, take a few minutes, read through the background, and then meet me back here. All right, cool. So if you're back here, you're through the background. Now, you're more familiar with the actual anatomy or the anatomical terms that are associated with a flowering plant. So now we're going to look at the materials. So we have our 4. So we have our daylily, which is in the tall one. We have our chrysanthemum flower, which is in the more stocky one. Then we have 2 tiny ones. We have the milkweed and we have the honeysuckle. Okay. In the box, you should have been provided forceps, a scalpel with a cap, scissors, which aren't used to cut necessarily. They're used to spread. In dissection, a wooden stick that isn't on your list. That's what I had in mind. And a bunch of dissection pins. So you can use these to kind of pin down the aspects of what you're dissecting. Things that I should also say, you got a pad, a foam tray, and a plastic bag. The plastic bag at the end is where you'll put all of the sample. and you seal it in the sample bag so you can, or the plastic bag, so you can dispose of it. Two things that they said were included in the materials kit, but were actually in the common kit are the magnifying glass and the ruler. So you have to go back to the common kit for those. And then other things are the paper towels. So just have like a stack of paper towels. And a camera to take pictures. A cell phone camera is perfectly fine. The only thing that I'll note is make sure that when you're touching your cell phone, you're not touching your cell phone with a gloved hand, since you're going to be touching, these plant specimens with your gloved hands, and they're plants. I get that. But, they're sitting in a preservative. You don't want that touching your phone. And then you're going to take your gloves off and you're going to wash your hands. And then you're going to touch your phone. And, so just don't do that. Safety, you want to going to be wearing your goggles, gloves, and apron. Yeah, so keep any open flames away from the preservative. They will catch fire. The absorbent pad, when you spread it out, you just want to make sure that you're spreading it absorbent pad blue side down. So the top has this white pad. That's what's actually going to absorb the bottom. If you do blue side up, whatever you spill on it's just going to kind of run off the side. And then the scalpel. All right. So important with scalpel care and how you use your scalpel. So never recap it. It's disposable. So once you take it off, it's off. If you have to recap it for any reason, do so very, very carefully. All right. When you go to uncap it, grab up at the top, grab at the base, and just pull straight. Don't put your hand here and pull. Right. Very simply. And when you're cutting with the scalpel, always make sure you're cutting away from whatever hand you have there. Don't cut towards your fingers. Don't cut towards your body. Cut away. All right. So all you need is one little slip or something, and you can cut yourself with the scalpel, and you could do some damage to yourself. And they have things here. Always use a sharp blade, which you should be because you always need this. Don't use a damaged scalpel, of course. Don't use the scalpel to cut through bone or heavy cartilage. You're not doing that here. Never reuse a disposable scalpel. That's important. Never cut towards your holding hand or your fingers. Always cut in a downward motion, right? So don't be trying to cut upwards. I don't know what you would be doing with that, but just don't do it. And then, yeah, the perfect solution, Carolina's perfect solution is the fixative or the preservative that they're using. Just try to avoid contact with your skin, definitely your eyes. Yeah, you don't want any of that in your eyes. Yeah, it can be a carcinogen. Yeah, so don't spill it on yourself if you can avoid it. But okay, so getting to the main dissection. So what I'm going to do is I'm going to set up my little area first and foremost. And then I'm going to get this all laid out. And we'll dissect some flowers. Not the most interesting dissection in the world, but important. Okay. So we have that. So let's see if I can angle this down and actually still be able to read what's going on. So you're just going to see my hand kind of move in front of the screen a little bit. Okay, so put on a new pair of gloves, put on your goggles. So we're going to be seeing my hands. So let's get some gloves on. Perfect area to do this is like a kitchen table. you live at home, preferably when your parents aren't around, so you don't get yelled at for dissecting things on the kitchen table. All right, take out the flask using, take each flower out of the flask using the forceps. Okay, we're going to put each flower then on a paper towel. With a new paper towel, we're going to carefully pat. Yeah, so, all right. So they don't outright state it, I don't think, but the foam tray is your dissection tray is essentially what they're calling it. So let's get all of our equipment up here that we're going to need. We don't have to be touching anything off of this. Okay, so we're going to go right onto the absorbing pad. Just a few paper towels. And pretty much what we're going to be doing, using our forceps, we're just going to be pulling them out and drying them off, but we don't want to crush them. Okay. You love the smell of fixative. Okay, and as soon as you're done, recap it. Okay. What I'm going to do is I'm just going to keep them in a particular order. That way I know what goes to where. Very very pungent smell. So I have no clue how you're supposed to get the milkweed flower out, because maybe with this. We'll get it. So my milkweed flower sunk all the way to the bottom. So I used this little wooden spear to kind of fish it up. So that might be what you need to do. The honeysuckle should be fine. All right, now hopefully your hands are still clean. Okay, now we just want to gently kind of try to get all excess preservative off. Without crushing. I'm just going to use, I have no idea what's going on with this plant. My box is kind of my trash can here. Okay. So using a clean hand. So at this point, take pictures of what you need. Okay, so we're going to pick up each flower, examine the exterior, note the position of the flower, the number of flowers per receptacle, the number per peduncle, may wish to refer to figures 4 to 7 at the end of the instructions to compare. Yeah, so we're definitely going to want to do that. Because as you see with preserved samples, they don't look quite the same. So first one we can look at is the chrysanthemum. Okay, so we have one peduncle, right? One big one coming up. We want to count the number of petals. Okay, so it's a little challenging to look at the number of petals because these petals are very thin. But we can do our best. So a technique that you can do is if you're trying to separate the petals, you can kind of push your forceps in between and then let them opening pull them apart rather than trying to grab and pull. So you can risk breaking. So it looks like we got like 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 24, 25. Again, it's going to be challenging. You know, don't kill yourself trying to figure this out. What's challenging with the chrysanthemum flower is, you know, it's kind of like in school when you have the in math class, you have like the real easy example on the board, and then you on the test, it's a really challenging one. So I'm guessing right in the middle is the let's see, it's challenging, right? Are all the ones in the middle the pestle? Go back up here real quick. Yeah, so let's see what we're actually doing again here. Yeah, number of flowers. So one flower per receptacle. Seems like that. Okay. Now we're going to look at the daylily. This we're going to try to open up the daylily. Mine is pretty closed. Hurting it. I'll see like a lily. A lily I know. Seen lilies. I know what lilies are supposed to look like. Oh, are these two? Okay. That's important. So at least on my lily, it looks like there's like a thin sheath. Okay. So you have like these darker ones on the outside. Then you have these thinner petals. Okay, we're getting towards the middle now. Right, and I think probably the lily is going to be the best one to deal with. Ideally, we would have two forceps here. Yep. We have to get into the middle of the lily to get to the stamens. Oh, I ripped a little on the flower. And there is the style. All right, I'm gonna stop there so I can see that. So I don't know if you can see it here, but right here we have the stamens on the lily. They look like these little bulbs. Try to get a little closer here. They look like these guys. I'll show better once I dissect. And then you have your pestle here, which is like a white thing. Okay, then we have our milkweed, which mine does not look very good, but let's see what we see. Again, this was the one that was kind of trapped at the bottom. Now, actually, this one's really cool once you get into it, because you can see the little stamens. And then you see the big, I guess, ovary right in the middle. And this one has two receptacles because it's kind of layered on top of each other. So that one's really cool. And then the honeysuckle is interesting because I don't know what is considered the stem on this. The way that my sample is, it's all sorts of messed up. But these little folded things are the flower. That's obvious. The stamens are obvious because they're like these little like T-bars that come up. I guess this is the actual the stem top of the base. That's the ovary. Okay. So okay. Carefully remove each petal with the scalpel. All right, compare the number, position, and size of the petals for each plant. Take a photograph of each flower. Record the data in table 1. Open the flower, spread it out carefully on a new piece of paper towel. Make sure not to damage the stamen or the pistils. All right, so now they actually want us to cut away the flowers or the petals, excuse me. All right. So let's do that. So we'll start with the lily. So again, be very careful. Does not take a lot to cut. The scalpels are very, very sharp. So you're just pulling and you're cutting. If you're struggling like I am, you can use your dissection pins, which I'm actually going to do. Put it right through the peduncle. That way it stops squirming all around. Okay. So now I've got to tease off these inner flowers. I feel like an easier assignment probably would have been just like go buy a fresh lily at like anywhere. So I'm just going to use the wooden stick to try to tease out the stamens here. It's kind of wrapped up in this petal. Without damaging them, which is going to be challenging because there's a ton in here. All right, take advantage of your pins here, everyone. If you have to. Okay. This one has all sorts of ones. But it's really cool because now that we've cut a little bit away, we can start to see the base, right? Where everything's kind of coming out of. Okay. Okay. Cool. All right. So just to show the daylily, got all the petals cut off. And what you can do for the instructions is for some reason they give you metal, everything else. I'm assuming this is just like a little probe, this wooden stick. Well, at least that's what I'm using it as. We can kind of separate out everything here. Okay. Cool. All right. So that is the daylily. So I'm just going to move that up to the front. Next is the chrysanthemum. This gets a little bit more challenging. Again, I'm going to pin. And for this, I'm going to just simply work with two hands. Just very slowly get the flowers off, or the petals off, excuse me. This one out of the way. Taking care of the petals on that is a lot less surgical than the lily, but effective either way. Okay, swarm the milkweed. So the issue that we're going to run into with these next two is that they are so small. But remember, let your scalpel do the work for you. Sharp. Let it do the work for you. Okay, now the challenging aspect, the milkweed, or seems like the inner flowers are really connected to the stamens. So I'm not going to cut those because I don't want to cut off all the stamens. And white on white is very hard to discern. Okay, last but not least is the honeysuckle. The challenging part with the honeysuckle, at least for me, are my honeysuckle's stamens are all tangled up. And it's hard to tell if any of these long ones without a cap are simply damaged, or well, that's how they're supposed to be. So when you're counting the number of stamens, right? So for instance, on the lily, you got 1, 2, 3, 4, 5. Mine has 5, has 1 pestle. Um, you know, on the milkweed, got 1, 2, 3, 4, 5, 6. It looks like 1 pistil. On the honeysuckle, what's challenging is you have 1, 2, 3, 4, 5, 6. If they're just damaged, it's hard to tell. And then on the chrysanthemum, I don't even know if we could count that. I'm not going to try. You can just put a lot. That's perfectly fine. Carefully remove the stamen with a scalpel. Examine the stamen with a magnifying glass. And so then examine the style, which is the tube of the pistil. So if we're looking at a magnifying glass, you can get pretty close. I don't know how much more detail you're going to see. I mean, maybe the chrysanthemum might be interesting. I don't really know how much more detail you're going to get with a little magnifying glass. Measure the length of each style. And measure the length of each ovary. All right, so challenging aspect is finding the ovaries. So let's start with the milkweed because that's the easiest one to me. So ovary length is about 1, 2, 3. So 0.3 centimeters. And the stamen length is for me about 0.5. The stamen length on the honeysuckle is about 2.3 centimeters. Stamen length on the daylily is about 4 centimeters. On the chrysanthemum, I got to be honest, I don't know. We'll look that up afterwards once I get my gloves off. Yeah, measure the length of each ovary. Select the flower with the largest ovary. Remove it. So what I'm going to do for this stage is I'm going to look specifically at the lily. I'm going to keep the milkweed to the side because that one's still pretty good. But this whole bulb aspect might be the ovary. So what I'm going to do is I want to get in there. And I want to see what's actually inside. Yeah, okay. Found the ovary of the daylily. How you would know to do that, I don't know. It's actually down past where the stamens actually form. Okay, and yes, on the daylily, the long one, like the long, same, looks the same, just missing the top part, the anther. That is the style, because you can trace that all the way down. Well, and that's much longer than we measured. Now for the ovary, it's challenging. So it's really kind of tightly, the tissue is very tightly wrapped around it. Getting in there without damaging it is challenging. It says to cut the ovary, right? Yeah. This is by far the largest ovary, FYI. I'm gonna move my dissection pins down a little. To help open up the ovary. I mean, oh yeah. Yeah, so you can see the little ovules in there. They look like clumps. So yeah, that's pretty cool. So then the daylily is the one that you want to be dissecting for the ovary. So you could cut it out if you wanted to. You could dissect it in there. Let's see, I could get pretty close. So if you cut down into this area, what you'll see is the stamens kind of tuck into the surrounding tissue. But the style, get that and pull that out without pulling out my pins of this lovely dissection dish that you were provided. Style, so you see here, it's the one without anything at the top. It doesn't have the anther, right? That traces all the way down to this little bulb structure, which I cut right through, which is the ovary. And inside you can kind of see these little lobes. Those are going to be your ovules. So the whole thing is the ovary, the little lobes are the ovules, and it'll look like a little bulb in here. But yeah, examine the style with a magnifying glass. You can see like the central canal. It'll run all the way down. But yeah, that is essentially this lab. You want to be taking pictures the whole time, doing things like that. But for the most part, that's the lab. So when you're done, you can unpin everything. So like I said, it definitely won't take an hour. Unpin, what I would recommend is use a single glove. Get that bag open. Throw all of your sample into said bag. It's all covered with even the flowers that you cut off. I would throw these also right into said bag. They have the preservative in them. And I would throw anything that you don't need also into said bag. So scalpel, anything that's not reusable, wooden stick. Paper towels that were used. Seal that up. Now things like ruler, forceps, that stuff, I would wipe them off with a Lysol wipe. That would be how I would work or clean them. Which we probably all have a ton of, thanks to the pandemic. We'll get that out of the way. There. You're gonna fold up your absorbent thing. Don't touch the absorbent side at all. Just roll it, fold it in upon itself. Like I said, I'm using a box as my trash bag. Go right into the trash can. All right. So the only thing that I was unsure of while doing this was the chrysanthemum. Don't judge me on my spelling. I wasn't even close. Let's see here. There we go. Chrysanthemum. Receptacle, ray, petals. It's not really. Oh, okay. Interesting. All right. So on the chrysanthemum, it seems like the ray is the so it looks like the ray flower, so one of these guys, has the stigma style ovary. So that would be female, right? And these are all kind of lined up. The disc has male and female. So it'll have the anther as well as the so it'll have the stamen as well as the pistil. That's interesting. So I don't really see a way in which you would have really gotten to do that in a preserved sample. Yeah. I mean, so like the taller ones within there. So they were the ones more in the middle. Those are purely, seems like the pistols. The ones on the outer edge would be the disc floored here, or the ones that have the stigma as well as the anther. So yeah. So pretty much what you can do is then you just kind of fill in number of petals, shape of flower, length of the stamen, number, length of the pistil, and number. For things like the chrysanthemum, kind of estimate style length and ovary length, again, for what you can see. Disposal and clean up, dispose of all waste into your household trash, but just make sure that you're taking caution. That's why I say, like, put it back in the box. You don't want your scalpel kind of piercing through a trash bag, or your wooden probe piercing through a trash bag. But yeah, so that's the end of this lab. If anybody has any questions, just let me know. All right, thanks everyone. Have a great rest of your night.